I'm going to give a talk on evolutionary biology and I guess in a plant breeding seminar series. And that's a real pleasure. It's a real treat. I looked at the list of speakers and I seem to stand out in that sense. But I hope that you'll find some point of contact. I hope that it'll be relevant. It turns out that maybe one of the two, I would say, singular realizations in plant evolutionary genomics in the last 15 years is something that's important to everybody in the room, that all plants have an evolutionary history with their genome has undergone repeated rounds of genome doubling. So my talk in that sense will be very general. The examples I'll draw will be from cotton, but they'll apply to all plants. And so this is cotton in its modern, highly annualized form. There's nothing like that in nature. But I prefer to start with a good place to start for all talks on evolutionary biology, which is, what's this guy's name? A little over 200 years ago, shown as a young man. And I'm getting old enough now where history is starting to become important. And so I like to think about things like, what would the impact be of the breathtaking technologies that we so enjoy today on questions that Darwin would have found to be centrally important. And you can just read these questions. They turn out to be timeless questions that also are very difficult to answer. What is the nature of the variation upon which natural selection acts? We still don't know. We're beginning to see more and more, though, aren't we, because of the technologies that we enjoy. So they are very difficult. They aged Darwin, as you can see here. But he was quite the astute gentleman, as there's the understatement of the day. He was much more interested in our cultivated plants and animals than he was in such famous organisms as the Galapagos finches. He spent a lot more time talking about sheep and oats than he did about wild animals and plants, for a very good reason. He said, it seemed to me probable that a careful study of cultivated plants would offer the best chance of making out this obscure problem. I may venture to express my conviction of the high value of such studies. And then he has this wonderful quote on cotton, which if I ever need a source of inspiration, of course, Charles Darwin is a pretty good one. And that's because he recognized that we have antecedent and descendant conditions still extant in many cases. They offer us a window into the evolutionary process that doesn't require thinking in deep time and the inaccessibility of long dead organisms and lineages. So this is a perspective that guides my work, and I'll share that with you today. And it'll be a talk in 3 sections. First, I'll give you a whirlwind tour of the genus Gossypium, a genus I fell in love with about 30 years ago. And it turns out to be a story involving Polyploidy, which, as I've already mentioned, is a story that is also applicable to all the plants in the room. And so I'll try to encapsulate about the last 15 years of work in 3 very short stories. One at the DNA level, one at the RNA level, and one having something to do with phenotypes and where we're headed. And again, using cotton as an example, I hope to illustrate general phenomena for polyploids in plants. And then I'll tie it up, as I said, with something about diversification and origin of form having to do with polyploids. Not just polyploids, but having to do with polyploids. Okay, so that last slide showed you cotton in its modern annualized form. It's a relatively recent immigrant to the United States, just in the last several hundred years, after a white man subjugated black man and took him halfway around the world so that we could grow this crop. Far more southerly antecedents, very important in the economy of the southern tier states and in one lab in central Iowa. And from this New World location, cotton spread worldwide, where it's the dominant fiber plant in the world today. One of the New World cultivated species, and yes, there's 4 different cultivated species, each derived from a different wild ancestor. And yes, that means we have this multiple parallel domestication that's taken place on opposite sides of the world, each by different cultures, each extending back about 6,000-7,000 years, probably, maybe more. So then this has now become the dominant textile plant in the world. Today it says here, cotton, one of Egypt's greatest treasures. This is a picture from a Land's End catalog about 20 years ago, so I keep using it in seminars. the reason I use it is I love it. Look in the background. It's right about when these were being built that this was being domesticated about half a world away. So this is the story of cotton in a nutshell. Photoperiod sensitive tropical antecedents in a van from Michael Gore's old haunt. This is Richard Percy's slide from Maricopa. This one's showing Gossypium barbadense during domestication, photoperiod sensitive tropical antecedents, modern high yielding annualized derivatives with a whole suite of architectural changes, yield changes, et cetera, et cetera. And people did this 4 times from 4 different ancestors. The trait that we're talking about, there's a whole bunch of plant biologists in the room, so I can say ovule, and I can say epidermal trichome, which looks like that on the day of anthesis. I don't have to explain anthesis, and that's what it looks like 2 months after pollination, which is days post-anthesis. That's what the DPA is. And the cotton fiber is nearly pure cellulose. It's over 95% cellulose, the secondary wall. And it forms this flattened spiraling ribbons. They'll stick together, and so they can be spun into yarns and threads. And that's why we're all wearing it. That's why it's somewhere in our bodies. Everybody is wearing cotton, almost certainly. Okay, but just like an ear of maize or a head of broccoli, there's nothing in nature that resembles that. The closest we get are plants that look like that, and humans did this from things that look like that. Here's wild species in the genus, and there's a tremendous range of variation. So that's what humans did. Ooh, isn't that a little disconcerting, that delay between the two? Is that bothering anybody? So wild species in the genus, and this I've spent a lot of time looking for them and collecting them, usually in the wintertime when it's fun to leave Iowa. I get to go to places like Australia, or find the cotton in this picture. See it, everybody? You're no longer, you can tell where we are. There's a hint. Right there, that's one of the wild D genome species. Here, see all the cotton in this picture? Yeah, right here. That 10-meter tall tree, that's Gossypium aridum in the western slopes of Mexico. How about this clade from tropical northwest Australia? These are herbaceous perennials that send out these massive underground, they have these massive underground rootstocks that send out these runners that'll bloom, and then they'll do a backflip when the capsule is mature, and they'll open up and they'll put these seeds right at ground level, which have repressed the trichomes. They just have this fat body instead, and the ants run off with them and plant them. It's the evolution it's actually a terminal clade of about 17 species in Gossypium, and it's fascinating because it's just the house that occupies the end of the road, where that road is extrafloral nectaries that exist throughout the genus, that the plant's hiring the ants as mercenaries for protection. Now they've just co-opted the whole system for seed dispersal as well. So it's wonderful. But when you collect them, you get thousands of little formic acid injections. So they're really viciously defended. Anyway, so there's this tremendous diversity. And what does a botanist do when he or she sees this kind of diversity? They say, I want to understand it. One of the tools we use is we build phylogenies. And this is what we've done for many years, and it looks like that. Although it didn't actually originate in Antarctica, we don't think. It's just I'm not good enough with PowerPoint to have it come out of the screen at you. And so we now recognize different monophyletic groups. Each of these genome groups is monophyletic. You may want to remember the A genome in Africa and the D genome in the New World, because those will figure prominently in today's talk. And we know about when it originated because we've sequenced genomes and all the genes. And there's a big error bar on this, but that's okay. We know the continents were largely in about the same place. We know they all have the same chromosome number, a haploid complement of 13 chromosomes, although they vary extraordinarily in genome size, as shown in the little call-out boxes here, reflecting, as you might expect, the TE component and different rates of proliferation and loss in different lineages, and that's a whole different topic. Notice the A genome is twice the size of the D genome. And we can diagnose the origin of certain kinds of morphology. So ask the question, when in the origin of the genus did long trichomes on the epidermis of the seed first evolve? And we know when it was. It was after the divergence of the F genome from the A genome. These are different scales. You can't see the bar there. That's 2 millimeters. That's a whole centimeter. So from those tiny seeds, that's the most recent common ancestor of the long-linted A genome cottons, which subsequently donated their long lint trait to all the New World cottons that dominate world cotton commerce today. Because the continents were largely in the same place as they are today, we can diagnose multiple transoceanic voyages. Just some of them that have taken place are diagrammed here, the most important of which is this one, where an A genome propagule is so important, I'll show it twice, because it's fun to do. where an A genome propagule crossed either the Atlantic or the Pacific, we don't know which. It took root. It then hybridized with a D genome plant, and we don't know whether in 1 stage, 2 stages, or 3 stages, somehow a polyploid lineage came out of that. And this is the origin of the New World allopolyploid cottons, which combine 2 genomes that differed twofold in size, an A and a D. And so when I first became interested in this 30 years ago, I wanted to solve this problem. I had this new bag of tricks, allozymes. And I was going to figure out who the parents were of the allopolyploids. And we didn't know when this happened. Maybe, there were proposals ranging, they thought maybe it was, continental drift that separated the previously sympatric clades into different continents with reciprocal extinction. Or others who thought maybe Kon-Tiki. Or in agricultural times, there was this neither. It's about 1 million or 2 years ago, these allopolyploids formed, and then they radiated into the species that we now recognize today, including the 2 that dominate cotton commerce, Gossypium hirsutum, that's the big one, and then this luxury one, Gossypium barbadense. You might have seen Pima cotton or Sea Island cotton. Your $200 shirts or sweaters, that comes from that one. Remarkably, and I say this, I really marvel at this, we have 2 new species of allopolyploid cotton. I think that's extraordinary given it's one of the most important genera on the planet, and so it's been studied forever. But we have 2 new species of allopolyploid cotton that we're in the process of writing up. Anyway, so again, they combine 2 genomes, an A and a D, and I just want to make sure I've made this point that polyploidy is ubiquitous in plants, it's recurring. We recognize sometimes auto and allopolyploids. That just describes the most recent event of genome doubling that happens during angiosperm evolution. In fact, all flowering plants have undergone multiple rounds of polyploidization. There's a deep literature on this subject now. So in the case of cotton, so all plants are polyploid. In the case of cotton, we finally finished sequencing one of the 2 diploid genomes. This came out last year, the D genome. The A genome has also been sequenced and is finished by 2 different groups, and there's a lot of resequencing going on for the allopolyploids. So we're finally getting the genomic tools we need to get at a lot of questions, and they're really enabling resources, these genome sequences. But what do we learn about allopolyploidy? Well, here's not cotton. Here's grapes, vitis, and that's supposed to be Theobroma cacao right there. And you can see that there's 3 different colors because they share this angiosperm triplication. And this is prior to the evolution of Gossypium. But then when tracing to Gossypium, there's an ancestral 5 or 6-fold multiplication. So what's 5 or 6 times 3? I didn't show at the bottom, which I could have right down here, 2 more duplications. So what's 2 times 2 times 6 times 3? And then I'm not talking about the A and D merger, which looks like that in my rainbow phylogeny. So now we have our neo-allopolyploid cotton. That's basically, who did the algebra? 100 and something, right? It was 144-fold, 144 ploid. Isn't that wonderful? And this basically applies to all of the plants that we work on. You can quibble about the numbers in the different lineages. In some, it's far fewer than in others. But isn't that great? Doesn't that help you appreciate how a plant genome can have 300 or 400 MIBs without invoking some special mechanism for their spread and colonization of a genome? I mean, simply by retention, lack of loss. I don't know, I think it's a neat perspective. But what it means is that these plants keep jettisoning all this extra DNA and then keeping some portion of it. And there's a lot of mysteries here and a lot of fun things to work on. Okay, here's an example of one chromosomal region in grape that is orthologous to one region in cacao, and you can see in this example 5 in cotton, and that's diploid cotton. But we're talking about the neo-allopolyploid cottons, this clade up here with Gossypium hirsutum and barbadense that shares an A genome and a D genome ancestor, again, that differ 2-fold in size. And so here's what they look like. There's one from Hawaii, one from Brazil, one from the Galapagos Islands. I love the name of this one, Gossypium darwinii. Isn't that nice? And this is Gossypium hirsutum. This is the wild form of the most important cotton. Here it is, growing in a salt spray zone in the northern part of the Yucatan Peninsula. And these are the forms that first attracted the attention of Aboriginal domesticators who brought them into domestication, kept selecting on them, ultimately leading to these dooryard forms that you can find throughout the tropics. You find a lot of feral cottons and a lot of these dooryard cottons that are maintained just sort of for lamp wicks or pillow stuffing, textiles, wound dressings, hygiene products, a great diversity of uses. And then later land races, and finally the annualized forms only in the last few hundred years that then subsequently spread worldwide, supplanting the vast majority of indigenous cotton cultivation in the old world. You may know about cotton in India and Pakistan and that part of the world, the two domesticated agenome diploids. There's little of that left. There's some. There's about 5% left still. But it's been supplanted by gossypium hirsutum, just because of its superior yield and fiber traits in most environments. Anyway, so this then, as I said, spread worldwide, as captured here beautifully in this picture from the Ivory Coast showing one tired worker resting on his work product, which this bale of cotton, which looks like a bowl of cotton in this book from Jan Arthus Bertrand's The Earth from Above. Wonderful coffee table book. Okay, that's your introduction to gossypium. That's the organismal side of the story. That's what we spent a long time trying to figure out and document, trying to understand this diversity. And now we know that this neo-allopolyploid contains two genomes, and we have that translates into the phylogeny you see on the right for any chromosome or pair of genes or gene expression or whatever. And we have a pretty reasonable understanding of the divergences involved. These are synonymous substitution rates based on sequencing about 10,000 genes. This is before we had relatively complete genome sequences. So now I'm going to tell you three short stories about polyploidy. And this all, I guess, was motivated by the idea that you like to think of your normal self-respecting diploid as having its 30,000 genes or 40,000 genes or, boring things like people that have only 27,000 genes. And they all work in this beautiful harmony to guide growth and development and reproduction. So how does allopolyploidy work, after all? You take two of these self-respecting diploids on opposite sides of the planet with a twofold different genome size, different ecologies, and you slam them back together in the same nucleus in only one of the two parental cytoplasms, by the way, agenome cytoplasm. All the New World cottons have Old World cytoplasm, mitochondrial and chloroplast DNAs. And it all works. And not only that, in some lineages, it can lead to new diversity. So that's just a fundamental mystery, right? So we started looking at it, and the first story I'll tell you is at the DNA level. And we're just trying to scratch the surface, and Jeff and Jane have done a lot of work in this area. Many of our colleagues around the country have done a tremendous amount of work in this area. And here's what we've learned, right there. And this is what he looks like. And we named him. We have the principle of priority holds. We get to name him. This is non-Mendel. And it looks like this. Here's the phylogenetic expectation that I showed you, and it turns out it's violated all over the place. There's lots of ways to violate it. You can get gene conversion or non-reciprocal homologous recombination. We don't have to get wrapped up in words. One copy overwrites the other copy. And we didn't know that could happen in allopolyploids. There's nothing but bivalence that we see. There's no evidence of any ephemeral contact between the chromosomes insofar as it has been looked at. But here's what it looks like. You've got a purple gene and a blue gene, the two homologs. You put them together in the same allopolyploid nucleus, and one of them can overwrite the other. Here they're both purple. Or you can get chimeric alleles, which is very interesting. You can actually increase the allelic diversity. You can decrease the nucleotide diversity. You can increase the diversity within one of the two genomes by decreasing the overall diversity with one copy overriding the other, because you're substituting one allele for the other. We thought that this would be rare. It happens in about 5% of the genes that we've looked at in the genome. We thought that probably just based on first principles, all of this action should take place early in allopolyploid formation when the two genomes are still learning to get along and there's a lot of mispairing. You can start mapping them phylogenetically. Here's just an example. So it's ongoing. It's probably even happening at the population level. We just haven't looked yet, but it'd be an easy thing to look at. What don't we know? We don't know anything else about the process. Does this have functional significance? Is it visible to selection? I mean, even when you overwrite an allele and you put it in, you're putting it in a different genomic context, right? It's not just the allele, it's not just the nucleotide sequence of the gene that matters, it's the genomic context in which it finds itself. It's a totally different expression environment, if you want, different promoters. So that's just one way in which non-Mendel has reared his ugly head, and it is an ugly head. I can actually say his ugly head. You get unequal rates of evolution, all the other ways you can imagine. You get gene loss. My colleague Josh Udall just showed that there's about 1,000 genes where presence-absence variation exists between the two diploids. And then what happens at the allopolyploid level, we're not quite sure. You get intergenomic transfer. Some transposable elements have gone a little crazy after allopolyploid formation, but not others. And that's the end of story one. And so all these phenomena have characterized each of these stages variously in each of the allopolyploids that we all work with to different degrees. Story two is at the RNA level, and you can tell I'm kind of zipping through these stories, because I have to. And this is an algebra story, and it's why 1 plus 1 does not equal to 2, and why 1 plus 1 divided by 2 is not even equal to 1. And so this, George Bush knew this, of course, that allopolyploids practice fuzzy math. And this first became apparent to us when Jeff Palmer sent his student Keith Adams to my lab to work on, as a postdoc, to work on gene expression. And this is now about, would be 13 years ago. And we had no idea about gene expression. We had no tools really to look at gene expression 13 years ago, except one gene at a time, by god-awful techniques such as single-stranded conformational polymorphism analysis. And who's done that in here? Raise your hand. Ugly work in the cold room, right, with sequencing gels. And can we all go have fun and wash sequencing plates together? Ooh, horrifying. Anyway, we decided to ask what happens to the duplicate copies of the genes. How are they used in an allopolyploid nucleus? Is it 50-50? Is it 50-50 in all cells? Is there cell level, organ level, tissue level partitioning? What goes on? We had no clue. So we just started one gene at a time. And here's an example. For one of the 7 genes that encodes alcohol dehydrogenase in diploid cotton, there's 14 in allopolyploid cotton. So a lot of ADH retention from all these past rounds of genome duplication, which is interesting in its own right. Anyway, this is an SSCP gel, and it just shows cDNAs run out on these basically sequencing gels, denaturing gels, and you show different organs up at the top and different levels of expression of the 2 homologs. And this is from one individual. And it goes crazy. It varies all over the map. So in leaves, there's 2/3 expression of the copy from the D genome and 1/3 from the A genome, and it's the reverse in roots, and you scan down, and look at that, you go, this nutty stuff. Alternate whorls of the same flower. You get reciprocal silencing of the alternative homolog. Got even sort of wackier when we started comparing that to what we see in synthetic allopolyploids, where here's a natural allopolyploid. Oh, I didn't even bother to tell you yet. We know when they formed, kind of. I mean, we know in quotes, 1 or 2 million years ago. So before people, long before agriculture, but not very long ago. We know that how, just from the molecular clock. We know how much divergence there is between the homologous copies throughout the genome in the allopolyploids. So we compare what happens in a 1,000,000-year-old allopolyploid to what happens in a synthetic allopolyploid. And these aren't exactly parallel lanes here, but let's see, stamen and carpel are the same, and they're the same. This is a synthetic. There's been no time for evolution. So we know that organ-specific silencing of homologs can be immediate. So we know it's an epigenetic phenomenon. So for those of you who are interested in polyploid theory, the gene duplication theory is all about time to subfunctionalization. And here what this suggests is time to subfunctionalization may be 0. In other words, simply just the epigenetic factors that control gene expression may actually silence a gene reciprocally in different tissues. We know this happens genome-wide now because we've continued it on with more high-throughput studies in subsequent years. So there's a lot of bias in the use of the two gene copies. We've spent a long time trying to figure this out just to get some understanding of the shape and scope of the phenomenon. And it's all over the map. So the gene, if you take any one gene, you can either have the blue copy or the green copy in any one tissue, and we call that complete or partial bias, or complete or partial bias in the other direction, A genome or D genome. And that happens, but there's also organ and tissue specific aggregate effects. So you can get any degree of bias among genes, and then it can be balanced with respect to the two parental genomes. It is modeled in the top leaf, where there's equal number of genes balanced blue and balanced green, or it can be unbalanced, where you can see there's a lot more blue being used than green. So there's sort of a differential usage of the two genomes, if you will, in some tissues. And we don't understand what causes this, or its functional implications, or its evolutionary significance, or anything you'd want to know. We're just still trying to describe the phenomenon. So here's an example just from the first, this is actually an ancient microarray experiment where we actually had to design homeolog-specific probes on a microarray, if you can imagine. So here's the results for the first 1,383 genes, and you can see there's a lot of D genome bias, more than there is A genome bias. If you look at the F1, 121 versus 301 genes. By the time you're 1 or 2 million years old, the overall level of bias has gone up. The use of the two genomes is more symmetrical. That seems to be a general rule. There's this temporal partitioning in the evolution of gene expression, and the two genomes learn to get along. Okay, it even happens during development of single cells. We've compared, we've looked at gene expression during development of wild gossypium hirsutum, which is from here, the north coast of the Yucatan Peninsula, in a modern domesticated form, and in parallel, wild Gossypium barbadense and domesticated Gossypium barbadense. And if you take a look through primary and secondary wall synthesis, you find at gene expression for homologous gene pairs, you find everything under the sun. Sometimes you find only one gene, one homolog use, sometimes the other. Sometimes you find 50-50 throughout development. That's not so uncommon. Sometimes you find only one and it increases or decreases. Sometimes you find complete replacement of one by the other, even during development of a single cell. So if you're thinking mechanistically, you're wondering how does that happen, right? Functionally, we have no idea, of course, either. So this is just setting the table for somebody else or for some future experiments. So biased homolog use is one lesson from allopolyploids, not just cotton. Everybody finds this no matter what you look at. And it varies tremendously by tissue, organ, situation. It's sensitive to rearing conditions. So is this second phenomenon, which is a little more challenging to understand. It was one that took us a long time to figure out. And it's called expression level dominance. That's one way to think that, so you can remember the name, or you can kind of think of it as, in some cases, duplicate gene pairs are co-regulated, and in other cases they're not. But that conclusion was revealed to us circuitously, and it goes something like this. If you take a look at gene expression in allopolyploid and you compare it to its two diploid parents, you see that the amount of differential expression between the allopolyploid and its two parents is asymmetric. This allopolyploid has total gene expression levels that mimic the D genome parent for most genes, where it's crazily different than the A genome parent, right? So it's asymmetric. 34 is not the same as 4. In fact, it's 8.5 times different. Well, you start looking at what this is at a gene by gene level, and before you start squinting at this slide, if you take three things, allopolyploid and two parents, there's only 12 non-trivial ways in which gene expression can be statistically different. One is trivial, it's all the same. But the other 12 are A can be higher or lower than D, and both can be statistically different than the allopolyploid. That allopolyploid can be higher or lower, that is transgressive to both parents, and so on and so forth. Well, these are the 12 categories. That's what these 12 cartoons are, and the little diagrams that you probably still can't see model the conditions. So in this one, the mom is downregulated relative to dad, and the allopolyploid is statistically different than both. And so that right there says 269. There are 269 genes that look like that. So here they are, mom, dad, both. Well, here's a category right here, 5,719 genes. This is an original microarray experiment. We've repeated it with RNA-seq. This came out maybe last year or the year before in Journal of Heredity. No, in you et al. What did we put that in? The New Phytologist? I don't remember. Anyway, so there's the expression of 5,719 genes in the A genome, in the D genome. So these are that set of 5,700 genes where D is upregulated relative to A. The allopolyploid looks just like the D. And it's not just for upregulation, it's also for downregulation. There's another 5,000 genes. So these are 10,000 genes where there's some sort of co-regulation. So it looks something like this. When you have D genome dominance, so that's what we're calling this, that the allopolyploid looks like the D, so it's D genome dominant. You get the A homolog is either up or down regulated relative to linearity. So they're being co-regulated, up or down. So we don't know why some gene, it works in both directions. We don't know why some gene pairs in these two genomes that differ twofold in size are up or down regulated and why some aren't. And you can imagine we're taking a look at that now. Obviously, it'd be wonderful if we had really robust allopolyploid genome sequences, which we don't yet have, but we're getting closer. We can have hundreds of thousands of little scaffolds if we want, but we're getting there. Okay, this is not just cotton. Nothing I'm telling you about is just cotton. Everybody who's looked at it finds it. It varies in tissues and directions. It evolves over time. The amount of co-regulation increases. Its bias in direction disappears. The two genomes seem to get along better. Everybody who's looking for it is finding it. It happens in Spartina, happens in coffee. Coffee, there's some really good work, a couple papers out now in allopolyploid coffee, showing its sensitivity to different growth chamber conditions. One mimicking one of the two parents' natural conditions more than the other. So this is this weird expression level dominance phenomenon. And it does reflect somehow initial conditions, and we don't know what its significance is. It's responsive to environment. Okay, that was story 2, the expression level. I think I'm doing okay on time. So story 3 is motivated by this picture. I said I'd say something about phenotypes. Look at these phenotypes. And when you look at those phenotypes, you can make a real obvious conclusion. The allopolyploid does not look like the mean of its two parents, right? Looks different. In fact, allopolyploid cultivars are capable of much longer, stronger, finer fiber than anything at the diploid level. I don't think I mentioned this, but they can be as big as individual Arabidopsis plants. It's kind of fun, but they're single cells and we can harvest them and we can get DNA and RNA and proteins and metabolites out of them. So we've moved on to doing proteomics and metabolomics. That's just underway. Published a little bit on the proteomics stuff. We're trying to figure out how you take 40,000 genes here and 40,000 genes here, and what happens to those 80,000 genes here in their developmental context to enable a longer and stronger and finer cell wall to be made or to be selected by humans. There's a good evolutionary problem and a real good challenge, and one that's, I would think, is of great interest to people, plant biologists of all stripes. Physiologists, we don't know how a plant makes a cell wall. This is a really good model to figure out how a plant makes a cell wall, for example, because we can study it developmentally. We can harvest this on the outside of the plant. You know, take a little liquid nitrogen, some glass beads, and you can isolate it. So we've started in some obvious places. It's another algebra story. Because we have these wild forms that look pretty crappy compared to their modern forms, we can do expression profiling. So we've done that using microarrays and RNA-seq, and we've moved on to doing proteomics, which is also interesting and gives a complementary and different picture. And so the experiments, you can imagine yourselves, they're really simple-minded. You grind them up, you do high throughput whatever, you start to compare, and you end up with huge gene lists. And as a guy with 3 plant taxonomy degrees, I look at the gene lists and I go, or can you please help me? Fortunately, I've got lots of nice colleagues who can help me, and there's some very interesting stories that come out of this. Let me just give you an example of what it looks like. Here's an RNA-seq experiment. This one is actually under review right now. If we block on wild versus domesticated. A nice picture there to show what you're doing. And you do lots of replicates of RNA-seq, and you then compare the results. You get something that looks like this. If I take 2 stages, 10 and 20 days post-anthesis, and I compare wild to domesticated cotton, so there's the number of differentially expressed genes in wild cotton between 10 and 20 days. So that means 2,122 genes were upregulated at 10 relative to 20, 2,478 genes were upregulated at 20 relative to 10. That's a lot of gene expression change, but it's a pretty dynamic period during the development of the cell. It's entering cell wall biosynthesis. It's concluding primary cell wall elongation. Cellulose synthase is rearing up big time. You're starting to put the brakes on elongation and start to make a secondary cell wall. And that's all, you can really mine these data for lots of interesting biology. I don't work at that level, but a lot of people are interested in that. You can do the same thing for domesticated cotton. The numbers are lower, which is intriguing. We think that's due to a sort of well, I won't even get into that. But that's not what an evolutionary biologist is interested in either, right? He or she is interested in that transition. And when you put in those numbers, you get, 4,000 genes. So 4,000 genes have been affected by human selection, at least at this transcriptional level. And you go, and that's just for 2 stages. If we look at 5 stages, it's about 12,000 genes. Actually, incidentally, I don't know that I've mentioned this yet. Probably about 3/4 of the gene in the genome have at least some modest level of gene expression during development of this cell. I mean, this cell is capturing, it's a really active and dynamic cell, tremendously variable. Well, you try to think of what you know about plant domestication. Think of famous domestication mutants. There aren't 4,000 lesions. I mean, humans haven't been around to select on 4,000 lesions. Think about, say, something like TB1, which we still don't know how it operates, remarkably. A little bit more with that hopscotch insertion 55, 60 kb upstream. But we don't know what it propagates through in terms of its whole transcriptional, translational, physiology, how it makes phenotype. And so we're trying to do that. We're trying to understand in a network sense how humans come along. Here's wild cotton. And they start practicing selection on just a few lesions. And since I had to work so hard on this, I'm going to show it again. Look at my mutations, right? And thank you. That made my day that you like that. And so somehow that's changed the shape. That could use some improvement, I admit. That's changed the shape of that whole network, which has impacted thousands of genes. It leads to a different phenotype. Okay, so we're now trying to figure this out. And in my lifetime, we're not going to get very far, but maybe we'll make some initial strides in this direction. So we're trying to use a battery of tools that we have at our disposal. And these are not unique to us. You're probably all using these too. For example, we're looking for the population genetic footprints of domestication. Selection winnows neutral genetic variation, right? So you take a neutral variation and you find the targets of selection. So that's one approach. And we're doing that in parallel because everything we do is in parallel because we've got one diploid, gossypium herbaceum, for which we have wild and domesticated representatives. The other one we don't have wild forms for, arboreum. No one's ever found it. But it's really clear that to me, it's really clear that it was independently domesticated. We've lost the wild ancestor of Gossypium arboreum. But anyway, so that's a diploid. Whoops. That's a diploid. These are the 2 allopolyploids. So we're trying to study domestication in parallel, all for their similar structure. And that's really fun to find what we mean by parallelism or convergence at each level of interrogation. Taking a QTL approach. This is with Michael Gore and Mi-Jong Yu and Josh Udall, a collaborative study. And then we're constructing these very tedious, this takes years, we're in about year 5 of just population development right now. Remember, these are long live perennials. Those wild plants, they may take a couple years to get your first flower. And then they'll give you one flower on December 12th. And then you'll wait for your next flower on December 26th, and you weren't there. Right? So this takes a long time to develop these, but we're developing these immortal near isogenic introgression lines. They'll be useful for everybody forever, and long after we're gone, because these are going to be single seed descended and collectively tile the whole genome, where we've recovered little bits of the donor parent and the recurrent parent, collectively tiling the whole genome, and then we can phenotype those in the field, and then hope to find the genes of interest, and then do all the different kinds of expression profiling that I've been talking about, or metabolome profiling, to try to dissect that complex architecture that underlies phenotype. So that's how we're going about it. There's probably all kinds of other ways we could do it, but NSF will only give you so much money, and there's only so much time, right? In the meantime, we've started to look at a couple other things just by staring at the gene lists and asking for help. So one of the genes that popped up, here's just an example. One of the genes that popped up in our comparative expression profiling was a gene that encoded something called profilin. Well, I wouldn't have known a profilin if it hit me in the head. But I've got friends who do. And so I talk to those friends. And they say, oh yeah, it works like this. You know, profilin helps shepherd actin around. So it's really important in cytoskeleton. Oh, that makes sense. Well, we said, okay, I wonder why it keeps showing up as upregulated by domestication. Well, first we didn't have any genome sequences. We better find out how many profilins there are in the genome and how many are expressed in fiber. And it turns out there's 5 in each diploid. 6 profilin genes. 5 are expressed in fiber. Ooh, then I wonder how many are expressed, how many of those potentially 10 homologs are expressed in the allopolyploids? Ooh, all 10. I wonder if humans have differentially affected each of these homologs. Like, and here's my secret hope. It was only the D genome homologs that were upregulated, right? So we've captured in action this old theory that polyploidy can give new traits by virtue of having one gene stay home, take care of business, and the other one free to do new and interesting things. So, but that's not the case. All the homologs were upregulated in all the species. So we clearly, blue is domesticated. What's that color? Lavender is wild in the 2 allopolyploids for each of the 5 genes in 3 parallel cases of domestication. So clearly what this means is that we have some sort of upstream regulatory factor that humans have unknowingly targeted. Doesn't even need to be the same one. Maybe we'll find that with these near isogenic introgression lines, for example. The other thing I hinted at is shown in this slide, where you have maybe recruitment of genes from the crummy genome. It's not a crummy genome, it's a perfectly nice genome, but it made crummy looking fiber from the standpoint of someone who wants to do weaving. Okay? So we're looking for genes that were targeted by domesticators without knowing it, post-polyploidization only for the D homolog. So here's an example. In hirsutum, the wild form, neither homolog has much expression. Whoops, 50% increase in this one. That's Gossypium darwinii from the Galapagos Islands. That's the wild form that we're using for Gossypium barbadense here. And under domestication, still nothing in the A homolog. The D homolog is upregulated. So this is a possible example of this recruitment of genes from the less favorable genome following allopolyploidization. That leads naturally to this quote, which I think I took from one of Jeff Doyle's many reviews on polyploidy. No, maybe Jeff's not quite that old. He's getting there. Well, I love this quote. I told you I'm getting more interested in history. So if as a consequence of polyploidy, a large number of genes become duplicated and the characters governed by such genes are of importance to the species, one of the members may mutate, leaving the character unimpaired, with the further possibility that mutation may be of benefit to the species. Isn't that wonderful? So Harlan wasn't the first person to say this, but he was really important. Long before Ono, whom all my zoologist friends still ascribe all the credit for divergence following duplication. But no, just go read everything S.G. Stevens ever wrote, for example, on polyploidy, a whole generation before Ono. But now we're getting to the point where actually maybe see some of this in action, huh? I mean, they could see some of it, with morphological markers. They just, when I started working on cotton, there were 67 known genes, 67 morphological markers. Now we're getting genome sequences. It's really exciting. Okay, I think I've finished in about the right amount of time. You mostly seem awake, which is good. Here's what I think I said, the top 10 list, evolution and polyploids. I tried to say something about the power of phylogenetics and genomics to reveal something about organismal history, which is necessary in comparative biology. There's a lot of non-independent evolution of duplicated genomes in allopolyploids. It takes a lot of different forms. The bias and silencing of duplicate genes in allopolyploids is everywhere, all allopolyploids. Gene expression in new allopolyploids globally can resemble one parent due to this co-regulation phenomenon, and we call it expression level dominance. Someday, maybe in a few years, we'll come up with much more insightful explanations for it and then better words for it, because it's probably a lot more than just one mechanism. It's probably a diversity of things going on under one sort of phenomenological observation. I didn't talk about transgressive expression. Expression partitioning of duplicate genes happens even during development of a single cell. Selection affects networks of thousands of genes, even when you're just talking about a single cell. So imagine, imagine how complicated evolutionary biology really is when you're talking about bigger, complicated phenotypes. We're talking about a single cell here. So there's still a lot to learn, huh? I mean, there's a lot of room in science for all of us and all of our descendants. Duplicate genes may be responsive to selection, polyploidy reduction, average crop improvement. So that's what I said. These are the people that did the work. And I thank you very much for your attention and for having me here.